Hello, everyone. My name is Esteban Galeano, and I work with Barb Thomas at the Department of Renewable Resources. Today, I will be talking about implementing cutting-edge molecular technologies in Alberta's tree breeding programs to increase genetic gain. So first of all, let's introduce the concept of tree breeding cycle. So how it works. First, we select parents from natural stands to establish the base population. After selecting, we perform breeding to establish the breeding population, and we test the families from the breeding population in progeny trials, and after testing, we select the best families and the best parents to establish the production population, which is called seed orchards. After the production, we can continue our cycle for a second or third generation orchards, and the goal is to maintain a balance between the genetic gain and to ensure diversity. I would like also to introduce the concept of pollen contamination. So let's imagine that we have our orchard in here, our seed orchard or the population, and we have in here a natural stand or another orchard from the same species, and unfortunately we can get contamination from pollen out of our orchard. This is a potential problem for most orchards around the world, and this can potentially decrease the genetic gain. Also, let's introduce the concept of genetic gain. It is obtained by selection, but it's affected by pollen contamination. So let's imagine a distribution of phenotypes in the parental generation with small trees in the population, with average trees, and with big trees. And these ones are the ones that will be selected as parents. In the next generation, we will have offspring produced from these parents, and they will configure a new distribution of phenotypes. We will have the phenotype from parents, which is corresponding to this average, and we will have a new population, which is the offsprings, with a better performance. The difference between the offspring and the average of the parents is called genetic gain. And the genetic gain is calculated by heritability multiplied by selection differential, which is what I explained, the difference between the average of the offsprings and the average of the population of the parents. And the genetic gain needs to be multiplied by the pollen contamination level. And this is how it's so important to have accurate pollen contamination levels. Because if we have 50% of pollen contamination, we can decrease half our genetic gain. And that's why we have been fostering the idea of genotyping different breeding programs in Alberta. Genotyping is the same as saying paternity testing, and how it works. Let's imagine that we have 8 trees that we want to genotype, and we want to know the relationship between them. So we collect the needles, we extract the DNA, and we will be using this SNP chip to understand the DNA and to understand the relations between them. The SNP chip will receive in each cell the DNA, and it is going to be placed in this equipment from Illumina company. After this, we will get this kind of diagrams called dendrograms to understand the grouping or the relationship between them. In here, you can see that tree 1, 2, and 3 are close related. Tree 6, 7, and 8 are close related, but tree number 5, it is not so related with the rest of them. This information can be used to measure diversity, calculating effective population sizes, and contributions of male and females. And also, we can use this information to increase gain by reconstructing pedigrees and measuring pollen contamination levels. So I would like to bring a case study, which is one of the white spruce breeding programs that we have been analyzing. It is called G1. We collected needles from founders close to Smoky Lake, seedlots from seed orchard in Grand Prairie, and progeny trials. And we asked these questions. How is the diversity in the different populations? How are the male and female contributions in the seedlots? And how is the pollen contamination and how this can affect the genetic gain? So we went to the founders and we collected the needles using pole pruners, and we went to the progeny trials, which have smaller trees, and we used scissors, and we bagged all the samples, and we kept them cold. So let's see some results. First of all, in here I am showing in blue columns the diversity, which is expressed in effective population size, and in the red dots I am showing the relatedness. The highest diversity was found in founders with the lowest relatedness, which is what we desire. And for seedlots 2003 and 9, we found a decrease in diversity and a slightly increase in relatedness. But after eliminating 65% of the trees in the orchard, and this elimination was based on performance from height of genotypes, we found a significant increase in relatedness and a significant decrease in diversity. The same with contributions. For 2003 and 2009 seedlots, we have balanced contributions from mothers and fathers, but after elimination of those genotypes, we found an unbalanced contribution. Let's see pollen contamination. What we found, different levels of pollen contamination from seedlots going from 50% down to 10%, and they correlated exponentially with wind direction. This correlation helped us to understand what was going on in the site. In these black squares, we can find other seed orchards that are not white spruce, and the white spruce ones are in here, and we found that the contamination source, it is located in here, and we have a significant pollen, external pollen coming from the target orchard of a study. Then we could reconstruct pedigrees with this genotyping technology, and don't panic with this graphic. I know that it's quite complex, but this is showing all the structure of the population with the females in colored dots, the males in black dots, and we can understand how the families are configured. And also we can find and understand the pollen contamination in seedlots and in progeny trials. This information can be used not only to find the pollen contamination, but also to get better estimates of genetic gains. So as conclusions, as our recommendations, first, the usual recommended distances between orchards from the same species or orchards and natural stands of 1 kilometer are insufficient to ensure low contamination levels. For this, we recommend to use drones carrying pollen. They are called drop copters. Second, random mating does not happen in operational tree breeding, so a careful design of breeding programs is needed. For this, we have different software, and I am just bringing an example as PopSync. Third, Tree breeders should avoid severe ruggings to prevent increases in relatedness and genetic diversity losses. One solution could be to collect pollen from the best males and pollinate the best females to get improved seed. And finally, we definitely recommend the adoption of genomic tools to advance the tree breeding programs in Alberta using SNP chips. So the perspectives for the different Alberta programs are the use of genotyping for accurate pollen contamination assessment, progeny testing and selection of parents, accurate effective population size calculations, understanding of contributions from male and females in seedlots, the first two ones to increase gain and the last two ones to ensure diversity to follow the public policies in Alberta and to have a balance between gain and diversity. I would like to thank our lab sponsors, all the team that have been helping us through all these years, and of course, the field's sponsors. Thank you very much again, and I am open to questions.